I guess, an underlying theme for what interests me the most in in biology and science in general, to look for simple underlying principles. And one of my favorite Picasso's paintings is this one that kind of illustrates that point, where, as you can tell here, he goes in this series of plates depicting a bull going from removing more and more detail until he gets to the essence of the bull. And that's sort of my scientific credo, that maybe not fully embraced in biology universally as it is embraced in math and physics and chemistry. And what I think is one of the sort of big problems in the way biologists, and I mean by that mostly experimental sort of mainstream biomedical science type of biology, is that we are addicted to complicated and devalue what's simple and often confuse it with what's simplistic. And in my definition, I'd say simple is when you get rid of anything that's not essential for understanding the problem. Simplistic is when you remove something that is important, and so it's you have to have the right intuition not to confuse it. And complicated is when you add things that are non-essential, and that's I think we're all experiencing that phase in medical research now, where there is too much of non-essential is added because methods are available to generate data. And so that's what often makes complex systems look complicated, whereas as you all know, the complex systems are not complicated. They have underlying simple rules. So what I chose to talk about today is one of the aspects, one aspect of biology that we're interested in the lab, which has to do with tissue biology and I would say cellular sort of sociology or social lives of cells. And because tissues are complex systems composed of multiple agents that are cells, they interact in some way and generate all kinds of properties that we try to understand. And the properties that we see, these are observables, and they don't tell us what the rules are that generated them. And that's sort of the main interest in that area for me. So to think about the notion of tissue, of course, is a very familiar. It's a textbook notion. It's very commonly used. We all kind of sort of know what we mean when we say tissues, but at the same time, they're kind of not well defined. If we try to give a definition to a cell, then we're fine. To the organ, to organ systems, we can give formal definitions, but for tissues, we actually can't. And all the available definitions they're quite ambiguous, as are distinctions between tissues, where one tissue stops and another starts, and so forth. So, and traditional classifications that divide them into four major types: epithelial, connective, muscle, and neuronal. It has a limited utility, mostly when we think in terms of development of organism, when we think in terms of pathology, in terms of any kind of biology that operates at the tissue level. We don't think of tissues as epithelial versus neuronal and so on. It's always, we think of it as a collection of cell types that belong to all these different types of origin. So that's a first problem. So we are kind of on a shaky ground to begin with, because we're dealing with something that's ill-defined. And that leads to these major gaps in our understanding of tissues. What are the patterns of cell organization within tissues? What aspects are pre-programmed versus emergent? And what are general themes and what are special cases? And that's again, something that I think biologists appreciate less than, let's say, physicists, distinguishing special cases from general cases. And that's what makes this whole area so interesting and challenging and fascinating to understand. To use tissue biology as sort of as an umbrella term for understanding, social organization of cells, to think of cells as agents in a complex system. And so this is to just illustrate how what exactly our gaps in understanding are. If we compare organismal biology, cell biology, and tissue biology, it's very clear that in tissue biology we are limited by understanding, not so much by data, but by understanding. So in organismal biology, we can tell that these animals that look drastically different, they're all variations on the theme of a mammal, right? And we can tell that there are some deviations from the common theme that we see in whales or shrews and so forth. But we can imagine a generic mammal with all the universal parts, all the organ systems. They all have central nervous system, 2 lungs, 2 kidneys, and so forth. And we know how they're functionally connected. And we can draw a generic, like a schema of a mammal. And then we can also say that a mammal is a special case of a vertebrate, which is more general schema and so forth. If we go to cells, the biology at cell level is the same. We can look at this, and these are dramatically different cells, dramatically different looking cells, especially if they were drawn to scale, it would be even more dramatic difference. And yet we know that this is all variation on a common theme of eukaryotic cell or mammalian cell shown here on the right. And we are not fooled by the fact that the most abundant and most easily accessible cell type, which is red blood cell, actually doesn't have a nucleus in mammals. We don't conclude from that that most cell types don't have nucleus, and therefore this is something essential and important for understanding. So we are not confused by that because we know enough cell biology to tell that this is a special case. And the reason I'm emphasizing this is because in here it's an obvious case of general versus special, but when we lack that understanding, we can be confused by looking at something that's most common, most observable, and we don't have the right intuition to tell if it's a general case or special case. So at tissue level, at the cell level, at organismal level, we have that type of knowledge. But if we look at the tissue level, there's different tissues, and then now we can't tell whether these tissues coming from different organs all represent variations on a theme. And what that generic theme is, and we cannot draw that generic tissue the way we can for generic cell or generic organism. And we don't know if there is one theme, and these are all some kind of deviations or variations on that theme, or there may be more than one theme, or maybe there are no common themes at all. But presumably there are common themes because all tissues have to solve the same set of problems. They have to solve a problem of having the right cell type in the right ratios, right quantities, right spatial arrangements, and so forth. They should types, not the others. And these are the same problems every tissue and every animal has to solve, and presumably it's been solved once in the beginning, and then everything was built on that solution. And therefore, we have a reason to think that there are general principles and general themes, and everything is some variations on these themes. And some of these cases may indeed be special cases. And that's what we want to understand, and that's what we currently don't understand. So instead we have, this is a picture like this, which is a textbook picture of what's supposed to be tissue. So the questions we want to understand is what are the rules that define, for example, cellular composition of tissues, how these different cell types know that they belong there in the right amounts, in the right proportions, in the right locations, and so forth. And if we approach it from the way that generally we approach something unknown, we try to kind of catalog things. We describe them by shapes, by whatever, some properties that we can measure. Nowadays, we do it by single cell analysis, by imaging, and so forth. And we generate this ever more complete catalog of characteristics. And that catalog doesn't bring us any closer to addressing the questions I mentioned in the previous slide. And there is a little doubt that having ever more complete description of cells and tissues using whatever next generation methodologies might be that may image every single molecule, let's say, and so forth, the amount of data will grow astronomically, and yet it's not going to bring us any closer to addressing the questions that I mentioned in the previous slide. And let's just use an analogy here for like, let's say if you're an alien and you're doing a PhD thesis on human social structure, you came to Earth and you look at people and you have these very powerful alien technologies that allow you to characterize everything you want about every human being. And you get their characteristics like names, height, weight, color of eyes, color of hair, insulin, glucose measurements, heart rate, and you catalog that increasingly. You know how many cells that person has versus that person has. And clearly that kind of data would be completely useless to understand what the human social structure is. On the other hand, if you approach it from a different perspective, you look at the human social group, and you start noticing that there's some ways that these different individuals relate each other that may define not identities, like identity characteristics, like name, height, weight, and so forth, but rather that some of them are parents and some of them are children, some of them are husbands and wives, some of them colleagues, some of them friends, some enemies, and so forth. And what all these words mean, these are categories. So there's a category of a parent, category of scientists, category of neighbors. But these categories defined, if we compare category of a parent or neighbor or friend versus a category of a good person or bad person, or a scientist by profession. So the profession category is still a cataloging category. It's just like the names and heights and whatever, body mass. Parent, friend, neighbor, these are categories that are defined by relations. And what I think is needed, we have the notion of a cell type, and we have increasingly detailed characteristics of cell types. And that's just like getting more and more data about individuals. But what we need is to go from cell types, which is a useful and fundamental notion, to the notion of cell categories. And the way they can be defined is just like the aliens would define social structure. They need to be defined in terms of relations. So just like relation parent-child provides deep insight into social structure, or relation friends or enemies and so forth, also provides deep insights that can explain social structure. Similarly, if we define cell relations, appropriate types of what would be the appropriate types of relations, that will give us insight into social organization of cells. And so what I think we need to do is to, rather than being obsessed with getting more and more granular data about, the identity characteristics, we need to define some categories based on relations. And so we need to ask, what are different cell types equivalent in some ways in terms of their roles in tissue organization? So that would define their relation, right? So if you have a teacher in one school, teacher in another school, they may teach different subjects, but they're defined by the relation of teacher and student. And if we know that relation, then we can say, okay, there's a category of teacher and there's a category of student. So what are the appropriate relevant categories for cells? And also, I would mention that not all relations are equally informative, right? So the category, some relations are more fundamental than others. So the same would apply to cells. And so we can define this, what would be called equivalence. So relations here, I mean, in more like in mathematical terms, like the way that relations are defined on sets. And they can be symmetric, reflexive, transitive, and so on. It's in that kind of spirit of the way that relations defines on sets. It's basically a way to partition set. So we have a set of cells, and what are different ways we can partition it? Just like we can take social group and partition it into parents, children, teachers, students, and so forth. And that, I think, is what is missing and what's limiting, that we don't have that kind of a perspective. And this is something we're actually working on, sort of from theory perspective. I'll just give a sort of a sense of what we're thinking now, because we don't have a complete list of that yet. So this is the sort of visual representation. What I mean, that we go from cell type paradigm, where we describe cell types, type A, B, C, and D, to cell relations paradigm, which will define cell categories. And here, for example, one relation will define between these two cell types, another relation between these two cell types, and so on. And importantly, this relation, let's say relation A, may apply not just for this epithelial and this mesenchymal cell. It may apply to other pairs of cells that belong to different types. And that's what's critical, because that's where we hit the wall when we try to understand general principle, and then we find, oh, macrophage is doing something here, so therefore macrophage as a cell type has this kind of a property. And then we don't find that example somewhere else and say, oh, oops, it's not generalizable. It may not be generalizable for cell type, but as a relation, it would be. So that's the general idea. And what are the types of relations? So I will just use a few examples to illustrate it. And the first example also deals with the notion of what is the general theme in tissue organization. And if we think about what's the simplest setup for an animal, we would see that all animals have epithelial mesenchymal units as the most fundamental, most ancient units of organization of tissues. There are no animals that don't have epithelial mesenchymal units. Even the simplest, like spongia and placozoa, that's basically, they pretty much don't have anything else, but that's what you have to have an animal. And they always have a particular type of arrangement. Epithelial cells, of course, form a polarized sheet. Mesenchymal cells are underneath, and they're separated by basement membrane. So that's a general structural setup. But what's interesting going beyond that is that there is a particular type of relation between mesenchyme and epithelium. And that relation in one instance is based on asymmetry in information transfer. So mesenchymal cells, for example, have positional information encoded by Hox genes. That translates into the choice of appropriate morphogen signals that they produce, for example, and then epithelial cells have several fate choices. And then mesenchymal cells, through this morphogen signal, dictate the fate choice that would be appropriate for a given positional information. So with this, we have this type of instructional relation here. And this is seen as a common theme in development, where mesenchyme will affect epithelium to shape it into different organs, different spatial structures, and so forth. And you can particularly well see it with development of different types of glands, which you can see in the next slide even better. We can say now you go from this generic organization of epithelium and mesenchyme. Mesenchyme is not shown here for simplicity. And then you can get all kinds of structures through a process of continuous transformation. So if you think about it as geometric transformation, it's an example of continuous transformation. There's nothing fundamentally new added here. All that happens is continuous transformation. These are all topologically invariant. And that means that by just using that simple module and just adding the variables that affect the degree of branching, you can generate all sorts of structures. And from a simple gut tube, you can go to making various types of glands. And ultimately, you can make pancreas and liver and lungs, and all by process of continuous transformation. So that's how powerful that effect of relation between mesenchyme and epithelium is. And of course, this goes back to Mangold and Spielman classical experiments of transplanting epidermis from one part of a bird to another part of the bird and observing that the appendages formed by epidermis are entirely defined by the underlying dermal structure, which is underlying mesenchyme. And from that came the notion that mesenchyme has some information about the map of the body, which we know is encoded by Hox genes, and that the epidermis or epithelium has several fate choices, in this case forming different kinds of appendages, feathers of different sorts, or scales, and that's dictated by mesenchyme. So now we take that, so here we're talking as biologists, mesenchyme, epidermis, feathers, and we abstract from that and say, okay, we have one type of cell, let's call it M cell, it doesn't have to be mesenchyme. We have another type of cell, let's call it E cell, doesn't have to be epithelial, and we define this as categories based on the relation between them, and the relation is as follows. M cell has some information, in one instance it will be positional information. E cell has some fate choices, in one instance this would be feathers and things like that. And M cell dictates the fate choice of E cell. And this we can generalize them and say that these are definitions of these two categories of cell that are based on the relation between them. And now we don't have to be attached to dermals, feathers, scales, and things like that. Now we can say there are two categories of cell. And in addition to dermal and epidermal, as a relation between M and E, we can have other types of biological scenarios. For example, M cell could be niche cell and E cell could be stem cell, because they also have the same relation. Niche cell defining fate choices of stem cells, self-renewal, differentiation, and so forth. Then we have relation in immunology between dendritic cell and T cells. So dendritic cell have some information about what kind of pathogen they encounter. Naive T cells, which is T cells that haven't been yet encountered any signals, they have fate choices. They can become different types of effector cells, memory cells, and so forth. What dictates that fate choice? Signals produced by dendritic cells. Because dendritic cells have that information, in this case, it's not positional information, but it's information they receive from encountering pathogens. And more generally, we can say for any control circuit, including homeostatic control circuits, sensors and effectors are two types of the circuit that where sensors has some information about whatever it monitors, and the effector can do something about it. It has some choices. Thermostat and AC, for example, like thermosensor knows what room temperature is. AC has options to increase or decrease, to turn on and off, and so forth. Same with beta cell and liver. Beta cell knows what glucose concentration, liver knows what to do about it. When beta cell tells it what to do through insulin. So this is a general relation. What are other possible types of relation? I'm not going to dwell on many of them, but I will just mention one or two more. So the second type is a functional relation. And this relates to a more familiar notion of division of labor. So here we have a classical textbook division of labor when two cell types specialize on performing two different functions, A and B, independently of each other. The second type of relation would be division of labor, would be when two cell types both contribute to the same function. And if you remove either of them, there is no function. So motor neuron and skeletal muscle form a motor unit, and you need both types of cells to have that function of movement. So they're functionally complementary. So that's now a type of relation. Here we can say this is also a relation, but it's a null relation. Here, it's a complementary, functional complementarity. The third type of relation that hasn't been described in literature, but I think is equally important, is a relation when one cell type is specialized on a particular function, another cell type is fully dedicated just supporting the function of the other cell. So example would be, for example, again, this could be motor neuron, and the supporting cell would be Schwann cell. So the whole purpose of life for Schwann cell is to support function of the neuron. Neuron is the one that performs primary function. Schwann cell just helps it to perform it better. There are neurons without Schwann cells. There are no Schwann cells without neurons. Schwann cell doesn't have meaning outside of neuron. It's like a golf player and caddy. You can play golf without a caddy, but caddy without golf player makes no sense. And that's how some cells are, like pericyte and endothelial cell, the same example, and so forth. So there are many cell types, particularly in more complex metazoans, that are of this type, where they perform supportive functions. And then other cell types perform primary functions. So here we have, again, a relation. It's like somebody who has, let's say, servants, like cooks and drivers, cleaners. There is that type of relation. So what happens is that the driver, cleaner, and cook, in this case, are defined by their relation to whoever they serve. And what that also illustrates is the way that this type of relation evolves is that when this cell type gets more and more specialized on particular function, what it means that when you specialize on something, it means you do fewer things better and better, right? Fewer and fewer things better and better. If you're like a famous, piano player and you don't have time to clean and shop and do chores, you hire people who do that for you. But what it means, you delegate some of the functions to supporting staff, supporting cells. So neuron delegates a lot of functions to glial cells, for example. And the same is true for all the other primary cell types. They can't be bothered to perform the same functions if you expect them to be optimal at performing one function. That's what happens with humans also. Of course, we get progressively more specialized, and in the process of that, we delegate some of the functions to other cells. And that in itself raises an interesting question: what can you delegate? So obviously, you can delegate functions that are not cell autonomous, but you cannot delegate cell autonomous functions. You can delegate cooking to somebody you hire to cook for you, but you cannot delegate eating, right? So certain types of functions are delegated, and these are non-cell autonomous functions. And the genes that perform these functions can now be expressed in cells that perform this supportive function. And that's what's seen in gene expression profiles and tissue-specific characteristics of cells that are ubiquitous in tissues, like macrophages and endothelial cells and so forth. So these are some other examples. I'm not going to dwell through all of them, but these are some other examples of cell relations that we can think of. I already mentioned instructive relation, functional relations. Then there could be supply-consumer relation, hierarchical relations, and there could be also relations based on mutual exclusivity. For example, if cell A is present somewhere, it's never in a company of cell B. These are mutually exclusive. We don't know, the examples like this exist, but we don't know if these are, they need to be defined in a more strict way. This is more just for illustration purposes here. Okay, so from here, we can now go back to this picture we had before, and now we can look at it in a very different light. We can say, okay, what do we have here? We have these various types of cells. They're not all equally important, because you can remove some of the cells and tissue will retain its architecture. You can remove T cells and you lose some functionalities, but you still have the same tissue architecture. You remove fibroblasts and it collapses. So they are unequal in that regard. And what that means is that some cells are more important than others for tissue architecture. They all have their distinct functionalities, but in terms of tissue architecture, they are not equal. And that raises the question, which ones are the most important ones, and so on. We can also now look at it from the perspective of structural relations, functional relations, and so on. So now, instead of being concerned with cell shapes and whatever, transcriptomics, proteomics, these sort of things, we can label them as different categories, and the patterns would start emerging, right? And so if you imagine them just as little circles with different colors, and we defined relations between colors, so the colors will represent categories. And so if we start thinking about what are the most fundamental categories and what are the most quote-unquote important cell types for tissue, for organization of tissues, not for functionalities, for organization of tissues. As I mentioned in the very beginning of metazoan life, it was epithelial mesenchymal unit with the basement membrane in between, and that's what led to diversification of various types of organization. In vertebrates, I think it's we have a minimal tissue unit that is based on that original theme. And that minimal unit consists of 4 types of cells, or 4 categories. 3 of them correspond to types, usually, for most organs, but these are categories. So we have a category of a tissue-specific cell that is responsible for core tissue function. This is the cell type responsible for the primary function of the tissue. In the brain, it would be neuron. In the gut, it would be gut epithelium, and so forth. And then we always have 3 additional cell types that are nearly universally present in all tissues, and that includes, of course, microvascular endothelium. It includes stromal cell, like fibroblast-like cells, and it includes tissue-resin macrophage. So these 3 correspond to cell types performing supportive functions. So now we define this minimal unit based on relation between these supportive cell types that are universal, they're in every tissue, and tissue-specific primary cell type that obviously is different in different tissues because it performs tissue-specific primary functions. Now, in the gut, we have, let's say, intestinal epithelium, then we have these 3 cell types. They are not involved in the primary function of the gut, which is digestion and absorption, but without these cell types, of course, epithelial cells cannot perform that function. In the brain, we have neurons and glial cells, the same idea. So the supportive cell types are always there to support the function of the client cell. So we can call this a client cell by analogy with chaperones. And so then what we have is that client cells delegate some of the functions to supportive cell types, and some delegated functions need to be performed all the time constitutively, and these become tissue-resident cells. But some functions only need to be performed on demand. And these are delegated to cells that are not resident in the tissue, but can be recruited to the tissue. And this is the case for vertebrates or any animal with close circulation. And that would be the case of inflammatory cells. So neutrophils, you only need them when there is an infection, whereas macrophage, you need in the tissue all the time because macrophages have many jobs. They're like butlers. They can do a lot of things, but when there is a really big problem, you need specialists, and that would be neutrophils. And so then we can think of the way that the logic of development of tissue architectures goes from this base case, which is what the beginning of animal life history, as well as the beginning of any animal during development, embryonic development. It starts with epithelium, then generate mesenchyme by AMT, and then from there you go through this process of instructive relations and generate all kinds of tissues. But the derived case that is universal theme now for vertebrate architecture, organization. This is only, we're talking about compositional organization, not spatial organization. The general theme would be that you always have a primary cell type, which is the primary function of tissue, it's tissue specific, and 3 supportive cell types. And that's what we can think of as a kind of first approximation. This is, of course, in very broad strokes, and this is only thinking about the compositional organization of tissue, not structural. But even from that perspective, there are many deep questions one can ask. We don't know the answers to most of them, but mostly I would say because the questions have not been asked that way. So one has to do with one of my sort of big interests for me is understanding biology of cell communication, and particularly biology of growth factors. So every cell in an animal, and every normal cell that is not cancerous cell, requires specific growth factors to stay alive and to be present in the tissue. The cells that don't have the specific growth factor will die by default. So cellular composition of tissue is dictated by local availability of these lineage restricted growth factors. For macrophage, it will be CSF1, from fibroblast PDGF-beta, and so forth. In endothelial cells will have VEGF and other angiogenic factors, and this cell will have different growth factor depending on what their identity is. So then the question becomes that the cellular composition, because it's a function of local availability of specific growth factors, what kind of cell, in what amounts, in what location you will have in the tissue is entirely dictated by local provision of growth factor in particular place in particular amounts. So then the question then becomes, what are the rules and mechanisms that ensure production of appropriate growth factors in the right amounts in the right places? And that, because we don't know those rules, I think that provides a major conceptual gap in our understanding of development and tissue biology. So we're interested in this question, and this is, of course, this is not one set of rules. It needs systematic study, and of course, in the lab we can only do, proof of concept studies. So the question is as follows: so the growth factors of that type, the type that are called lineage restricted growth factors that are necessary for each cell to survive. Production of these growth factors is typically the source of them is another cell type. If this is growth factor for cell Y, it's typically produced by cell X, which is a different cell type. So cell X makes growth factor for cell Y, and the amount of that growth factor determines the number of cell Ys, where they are, location, because these growth factors typically have small diffusion radius, and they are often deposited on ECM. And then the question becomes, how does these cell X knows which growth factor to make and how much to make? Okay, we can say, well, which growth factor to make could be hardwired. It could be part of the identity, that if you cell X, you program to make growth factor for cell Y. That's fair assumption, and there are cases when it is indeed the case, that it already knows which growth factor to make. In other cases, it needs to be told. And it can be told by cell Y itself. Cell Y tells it that I need, my identity is Y, and I need growth factor that supports cell Y. And what that communication is like, that's the part that we also interested in understanding, and we still don't know all the details, but we know that there is indeed this type of communication. So the question is, what determines this, controls the appropriate production level. And I'll go through this example very quickly because it was published, but we use macrophage fibroblasts as a case study as a proof of concept study to characterize in exchange of these two growth factors, which are well defined for these cells. It's CSF1 for macrophage produced by fibroblast, and PDGF for fibroblast produced by macrophage. And what we found in that study a few years ago is that if we define conditions such that if you take individually this cell type and don't provide exogenous recombinant growth factors, they will die as expected. Each of them will die. But because they can each produce growth factor for the other guy, we ask whether they can support each other under those conditions where there is no external growth factor. And they can indeed, and not only that, they could also reach stable ratio, even if we vary initial ratio by 100-fold. They all converge on stable ratio, which implies that they're not simply feeding each other growth factor that would lead to uncontrolled growth, but rather there is some kind of a circuit design of the exchange of growth factor that results in stability. And indeed, there is a circuit design that results in stability, and it's shown here. So that's what we found both experimentally and through modeling, that for the two cell types to be stable, one has to be limited by carrying capacity, which is limitation external to the system, just like in ecology, carrying capacity, space, food, and so forth. And the other cell type has to be limited. It has to have a negative feedback on the growth factor it receives from the first cell type. And as long as these two conditions are met, you will have a stable system. But these conditions are met only in 24 out of 144 total cases of total number of possible cell circuit topologies. Of them, only 24 are stable, and all of them have these two conditions met. And this is what it would look like if X1 is here, would be fibroblast, X2 macrophage. This is a phase portrait of the dynamic behavior. We have the same as experimental data. And this is the predicted on state, which is what we observe. And as you can see, the system will evolve towards the stable on state, and this is stable off state when there are no cells. But what was interesting in this case, we also found additional on state, stable on state for one cell type, for fibroblasts, but only when the density of this cell was above some threshold. And in ecology, this type of phenomenon is known as Ali effect. Ali effect means that population will undergo expansion only if it is above some critical density. So, for example, emperor penguins, they cannot survive in the wild if there is not enough of them, because they need to huddle to keep each other warm. Pack of wolves, one wolf would not survive because they hunt in packs and things like that. So this is Ali effect, and we see that there is Ali effect in cell ecology as well. And specifically for fibroblasts, it requires, if they reach critical density, they now can support each other without macrophage. And the reason for that is this autocrine signal. And that signal is provided by PTGF alpha and HBGF. And so that's what type of scenarios we see, that for this is stable observed state. With autocrine signal, you have this additional stable state that could be pathological, because when you have fibroblasts without macrophage, this is basically the case of fibrosis. So this type of circuit design creates vulnerability for fibrosis. But then there are other designs that we find through computationally, and they presumably exist also amongst cell-cell interactions. In this case, if you don't have autocrine signal, you don't have this pathological stable state with cell X1 alone. But on the other hand, so these type of tissues that have these cells with this circuit of interaction will not be vulnerable to fibrosis. But on the other hand, the basin of attraction to the off state is much bigger here, which may suggest that these type of tissues will be vulnerable to degenerative diseases instead of fibrotic diseases. And this is something that we're still exploring if that's indeed the case. So from that, what we find is that there is this relation between cells based on growth factor production that is dictated by particular growth factor topology, and that this there is a simple sort of requirement for stability that we can define, and that this can be generalized to other types of interactions where it's the same topology present. And from that, we then wanted to ask the next question: what controls the size of the compartments in tissues? And there are two paradigms that existed side by side for decades. One paradigm for compartment size control is basically space availability, and that cells, if they have cells to expand to, they will expand. When they don't have cell space to expand to, they stop expanding. This is contact inhibition in vitro, but in vivo, this would apply to cells that use mechanosensing, for example, to determine when they have to stop dividing. And this type of mechanosensing, for example, famously operates through the Hippo pathway. And that paradigm works for these types of tissues, but it would not be applicable to tissues that are not limited by space, like macrophages, lymphocytes, and a hematopoietic cell. So then the question is, what controls their compartment size? And that's where a second paradigm is applicable, and that is that every cell needs growth factor to survive and to proliferate. And if you fix the amount of growth factor to the appropriate level, the cells will expand if there is more growth factor than the cells that it can support. But once they reach that level, further expansion will result cells that now don't have enough growth factor and they will die. And so it's above that level, it will go down. If it's below that level, they will expand. And that's how you control. And that clearly that's how it works for hematopoietic cells. That's well known. You can change the amount of erythropoietin, for example, or GCSF or IL-7, and you can control different hematopoietic cell size numbers. But what was striking for us is that these two paradigms applied exactly to the two cell types that we use in our study. And again, if we think from the cell category perspective, we have one category of cell that is limited by one population size control paradigm, and the other cell type that's controlled by another paradigm. And we asked whether this is just purely a lucky coincidence or curious coincidence, or if there is some deeper insight here. And specifically, we asked whether, because fibroblasts controlled by space availability, so carrying capacity of the environment, and macrophage by growth factor availability, we asked whether production of growth factor by fibroblasts is a function of space availability it can detect. And that's exactly what we found, that when we looked at fibroblasts, growing them at different densities as to mimic space availability, we find there's a number of genes that are controlled in density dependent manner, and amongst them growth factors like CSF1 and many others. And so that indicated that CSF1, for example, would be expressed at higher level when cells are at lower density. And as density increases, it will be at lower level. And this way you adjust the numbers of fibroblasts dictated by space availability to the number of macrophages dictated by CSF1 availability that they produce as a function of space.
I have a quick question. Sorry to interrupt you,
Sure.
but so I know that the ECM plays a large role in growth factor availability. It acts like a sink. It acts as like a release factor. Have you looked at all at, because, you know, in cell density, you start to build up a certain amount of ECM as well as the number of cells. Have you looked at whether or not if you cotemplate with the ECM that these cells would normally lay down, if that would have the same effect as the cell density?
Yes, we have. The answer is a bit more kind of involved it's a very important question, and we really interested in understanding how ECM contributes to that. We know that it's a little bit, it will take us a bit far from the main topic here, but what we know is that in the case of macrophage and fibroblast interaction, the exchange of growth factors is cell contact dependent. Moreover, fibroblasts that receive PDGF from macrophage will only receive PDGF from macrophage if it provides that macrophage with CSF1. So if we remove CSF1 from half of fibroblasts, mix them with the wild-type ones, give them macrophages in the same system I described, fibroblasts that don't contribute growth factor do not receive growth factor. So it operates in a cell type. It's like gangster movies where you give the drug and the case with drug and case with money, and you exchange it like this, and in contact you don't just give and trust the other one. So it's some kind of a maybe anti-cheater strategy, but it is a direct exchange in this case. There are some other very interesting scenarios, as you mentioned, because there is a form of CSF1 that is a proteoglycan form. But in this case, it wasn't a factor. We tried to simplify it to look at from the with a minimal number of variables, but it is a very important point. There is a mechanosensing by both fibroblasts and macrophages, also a thing that I'm not going to discuss, but where we find that macrophages use very fundamentally different mechanosensing strategy compared to fibroblasts or epithelial cells. Sorry, so just to finish this quickly, this part, what we find is that there is a density sensitive enhancer in fibroblasts that controls expression CSF1. And when we perform motif enrichment analysis, we found that TED sites were enriched in density sensitive genes, and TED is DNA binding transcription factor controlled by Hippo pathway. And basically we found that Hippo pathway controls, through this distant enhancer, controls expression of CSF1 in fibroblasts, but not in other cell types. This enhancer only active in fibroblasts, and TED1 indeed is recruited there, as shown here by chromatin IP, and that's what controls expression of CSF1, leading to this kind of a model where we have homeostatic or density sensitive enhancer in CSF1 gene that is operative in fibroblasts, and it controls homeostatic expression of CSF1, where it adjusts the number of macrophages to number of fibroblasts. What the parameter for the ratio, this is exactly the kind of characteristics that would be dependent on the tissue and properties of ECM and so on. That's where the tuning of this parameter will change this ratio in a tissue-specific way, because not every tissue has exact same ratio and doesn't need to, and should not have. And then there is this, the inducible enhancer in FKB dependent that allows for expansion of macrophage on demand during infection, inflammation, and so on. So that's the picture we see with this type of relation of macrophage and fibroblasts establishing appropriate cell numbers. And of course, this is just one of the principles. There are many additional, as mentioned, ECM, how they both sense ECM, and whether signals is delivered through cell contact, through diffusion, or through deposition on ECM. This will provide important variations on the theme, which need to be individually explored. But what I want to finish here is with the notion that there is how this can be generalized, where we have a cell type, for example, fibroblast, that senses, for some aspect of tissue environment, for example, space. That space sensing is coupled, in this case, through Hippo pathway, coupled with control of growth factor for another cell type. And because space availability, cell numbers, these are characteristics of tissue homeostasis, it's homeostatic variables. Here we just have this simple type of relation, unrelated to function, because space availability is not related to function of macrophage. But then we have other types of relations where it is related to function. So for example, sensing oxygen availability, another environmental factor in tissues, now sensed through HIF1 pathway, and negatively regulates expression of VEGF, which is a growth factor for endothelial cell. Now, endothelial cell functionally is related to oxygen availability. So if oxygen level is low, it's derepressed, angiogenesis occurs, oxygen delivered, and oxygen level goes back to normal. And that type of relation now can be expanded, as I will elaborate in the next slide. But there are additional types. So this raises the question of what are aspects of tissue environment that are sensed, all the aspects of tissue environment that are sensed in a way that translates into control of specific growth factor production, and how that, because each growth factor controls specific cell types, how the cell type is related to this factor that is sensed, that controls production. And that, I think, what defines the rules. It's one set of rules. It's not all rules, but it's one set of rules that defines how growth factor expression is regulated. So here we have the homeostatic control, which is unrelated to this functional role of the cells. It's calibrated. You always need enough macrophages. So it's calibrated on evolution timescale. You always need this many, this ratio, this percentage of macrophages. But then, in addition, you can have expansion on demand. If there's infection, you need more macrophages, so this growth factor will go up. If you have hypoxia, you will need more endothelial cells, this growth factor will go up. If there is this X, some other environmental factor of tissue, you need more of cell X, and you produce growth factor for cell X. And then when we looked at imposing different kind of perturbations on cell to mimic these deviations in tissue environment. So we used hypoxia, glucose deprivation, amino acid deprivation, ER stress, osmotic stress, and so forth, oxidative stress, and then looked at the expression of genes in fibroblasts, and we find a whole range of growth factors are regulated by these perturbations, which brings up the possibility of exploring what the logic is in each individual case, because for each growth factor, there is a corresponding cell type that's controlled by it. Often for a given perturbation, you will need more than one cell type, of course, and that's why you will see that some of them induce, most of them induce more than one growth factor. So sensing of the environment by one cell type is linked to control of population size of another cell type that is functionally related to whatever that environmental perturbation reflects. And so to generalize this, we can say X is anything that determines the requirement for Y. Let's say there is a lot of, I don't know, garbage, you need more janitors, right? And then you have a sensor that determines that there is a lot of garbage. So that would be the cell that determines that X is there, and therefore you need Y to take care of X. If it's hypoxia, you need endothelial cells. If it's infection, you need neutrophil. If it's some kind of other type of perturbation, you need a particular cell type. And then the way that it operates is what I call functional demand. So in this case, what the underlying logic is that if X is detected, that means there is a demand for the function of the cell Y that can take care of X. So this is a sensing of functional demand by cell X. So it sends demand for function of cell Y, produces growth factor for cell Y expands, performs the function, functional demand is eliminated, and it goes back to steady state. And in this fashion, cell X knows which growth factor to make. Remember the question of identity, how do cells know which growth factor? It knows which growth factor to make, and it knows how much to make. It depends on how much of functional demand there is. And it doesn't have to be only growth factor, it could be any signal that can fulfill this circuit. It could be expansion of the cells, but it could also be recruitment, or it could be activation of the function Y that will remove that demand. And that now goes to a more general type of control circuits that operate also in physiological settings and so forth. But for growth factor production, I think that's what determines their functionality. Okay, so I will, instead of continuing in the same way, what I will do, I wanted to finish with something very not different, but with notions related to what I started with, thinking about what I talked about. Most of my talk is about examples of approaching these questions of tissue, social organization of tissues, social lives of cells from the perspective of trying to look for underlying principles and rules. And of course, as you all know, this is the general sort of strategy and general paradigm in complex systems where you have multiple agents that interact with each other according to some rules, and that generates various emerging properties that are consequences of these interactions. And of course, then the pictures like this that are emergent properties of interactions of agents, fish or birds, these are the observables. And the observables often fascinating, but understanding them from the perspective that would be the first instinct to understand them, to describe the size, the number of fish, and what they're thinking, what their glucose level is, measuring their RNA expression levels and proteomics and so forth. these are going to be totally useless to understanding how you form this type of structures. And instead, we need to understand what are the rules that generated them. But again, what we're dealing with is these observables. And I made this very kind of a toy example to illustrate something that I think for you would be very familiar intellectually. That, let's say this is the observable, this is what you see in microscope or through some other type of experiment. And you try to understand how is this generated. This is some kind of a structure you see. It could be some arrangement of cells in the tissue or whatnot. And of course, this would be very misleading if you try to kind of use your immediate instinct to think that there is something that generates a curved structure, right? Then if we know that actually it's part of this structure, it's just that you can see there are 4 repeated units and 3 of them are black and you cannot see them, or 3 of them are made up by cells that died as a part of their developmental program, and you're left only with this one on the left. But in reality, it's actually symmetrical structure. Not only it's curved, but it's still curved. So it's still thinking, well, how do you generate curved structure? And then we unmask additional layer, and now we look here, and now we see that, these are actually curved structure. Now the white stripes become visible. And once they are visible, now we see, oh, this is just slightly skewed hexagon. And we know exactly how to make a hexagon. And this is just one line of code to say, draw a hexagon, and instead of 120 degrees, you turn by 121 degrees. And now it's slightly skewed, and you iterate that and you get curved structure. And if you remove some parts of it and end up with what's on the left, then there is no way to tell how you made it. So the point being that the rule to generate this is very simple rule, just drawing hexagon and slightly skewing the angle. As a result, you have these spiral-like structures, then you remove parts of them and you can have a curve. And if you look at the curve, it's very hard to tell, if not impossible to tell, how it was generated. And so what we deal with often is these observables, which are several steps removed from the rules that generated them. The rules themselves are extremely simple, but the observable, the curve, is the emergent property here visually, but how it's generated is unclear. And we can see that more clearly with some cell automata types of examples, where you look at this final structure here, and you try to understand how it was generated. It's hard to tell by looking at it, but that's the observable. Now, if you look at it carefully, you start seeing, well, there is some symmetry here. And if you look at symmetry, you apply coarse graining and see that, well, this unit actually repeated 4 times, this one. In this unit, you also start seeing symmetry. It's also repeated 4 times, and in this structure, then it's repeated 4 times in this structure. And that is generated by these types of rules. So these are 2 rules. These are completely randomly chosen just for illustration purposes to say that these are the rules that generate this. The way they generate is through iteration, and based on local information only, and based on only information available at the time of execution of the rule. You don't need to have memory, you don't need to have global information. But the point is that if we know the rules, of course, we can generate easily these structures, and we understand them completely when we know the rules. But if you go the other way, which is the way we're usually forced to go, because what's observable is going from the other end, we have this structure, and we try to understand the rule, and we can't because we don't know. There are no universal methods to do so. So we have to, instead of characterizing in more and more detail what that is, we need to try to figure out what are the original structures that generated them. And this is what basically point that what we need to understand is what are the rules of interaction. And of course, then there are many fascinating questions about specific mechanisms, pathways, and so forth. But if we simplify it down to the bare minimum, if we think about what cells can do 5 things. They can do nothing, so that's an identity operation. They can die, they can copy, they can change identity, and they can change location. Sorry, I said identity twice. I meant it in different ways. This is cell identity, and this is identity operation, meaning that it's unity. It's like doing nothing. So these is 5 things that cells can do. There's nothing else. Everything is variation on these 5 things. Of course, most variations comes from changing identity, which is cell state, cell type, and so forth. But ultimately, everything is a combination of these 5 actions. And this first action, do nothing, is also an active action because you don't just stay as you are. You often, a cell always needs a signal that tells it to stay as you are. Don't die, don't proliferate, don't move. It's also an active process. So for each of these, there is a corresponding types of signals that tell them do A, B, C, D, or E. And so now if you think of all the language of cell communication, all the words that cells can hear and utter, all of them, we can say, can fall into 5 categories, whether they tell cells to do A, B, C, D, or E. And that is sort of the direction we are going in our thinking of defining the language based on, of course, the interpretation of different signals also depends, also a function of the state. It's not just that a particular biochemical signal always means A or B. It also depends on cell state, just like in any communication. And those are the very basic and general sort of description of the types of rules we're seeking to find. So I'll stop here and take any questions. Thank you.